As recently as 1981, if you had diabetes, your life depended on insulin harvested from the pancreases of cows and pigs. It took around 30 animals a year to keep one person alive. But that all changed in 1982. Scientists figured out how to get bacteria to make insulin that was chemically identical to human insulin. It became the first FDA-approved drug to use a new genetic engineering technology, and it marked the birth of the modern biotech industry. But here's the twist. Despite being a revolutionary new medicine, human insulin isn't that popular anymore. In 2018, over 90% of insulin products used in the United States weren't pig or human insulins, but something else. Something we needed genetic engineering in order to create. So in today's video, we'll learn how scientists came up with this new technology and used it to create human insulin. Then we'll take a closer look at how biotech started working with pharmaceutical companies, which set the standard for these partnerships in modern day. And finally, we'll learn how biotech gave us that new, popular insulin product. And oh boy, does this story start off with a bang. In 1972, a team of biologists led by Paul Berg at Stanford University published a massively important paper. They had come up with a method for attaching two bits of DNA from two separate organisms. This was later called recombinant DNA, because they had recombined DNA. But the process was complicated. It took a bunch of different enzymes to reattach the two pieces of DNA. Luckily, a lab just a few miles away from Stanford had a tool that would make the process a lot easier. The lab was led by Herb Boyer, a fun-loving, mustachioed professor at the University of California, San Francisco. He had a special enzyme that could cut DNA at specific, predictable locations. It was called a restriction enzyme. One of Boyer's students isolated one of these enzymes from an E. coli bacteria, so it was called E. coli restriction endonuclease 1, or EcoR1 for short. And soon afterwards, Stanford researchers found something unexpected. The EcoR1 enzyme not only cut DNA in predictable locations, they also left these little overhangs of genetic material on one end with mirror opposite overhangs on the other end. And this was significant because sections of DNA pair extremely predictably. C pairs with G and A pairs with T. So these tips, what they called sticky ends, reattached on their own because they could hook up to the matching sequence on the other sticky end. And on the practical level, sticky ends made it possible to combine genes that contained the mirrored sticky ends, which was anything cut by the EcoR1 enzyme. So all in all, these restriction enzymes made it much easier to make recombinant DNA. Now, one of the guys keeping up with this research was a Stanford scientist doctor named Stanley Cohen. He was studying circular pieces of bacterial DNA called plasmids. These things allow bacteria to transfer genes to other existing bacteria who can then pass that gene on to their offspring. And Cohen was particularly interested in PSC101, a plasmid that carried a gene which gave bacteria resistance to certain antibiotics. Up to that point, he'd been breaking up plasmids in a literal blender, which obviously doesn't make for super precise cuts. So this restriction enzyme would be useful in his own work, but Cohen also wondered if he could use recombinant DNA to stick new genes into plasmids. And if he could do that, maybe he could put that new plasmid into a bacterium. So Cohen met up with Boyer at a conference in Hawaii in November of 1972. They hit it off and decided to work on a project together in the new year. Cohen would bring his plasmid expertise and Boyer would bring his restriction enzymes. Biologists call this an absolute party. On the day-to-day, they'd cut up Cohen's plasmid with the EcoR1 enzyme, then cut up a different plasmid and combine the two. From there, they would check their work using a process called gel electrophoresis. This process separates fragments of DNA by size, so smaller pieces of DNA travel further through this little gel while big pieces stay close to the starting line. It's a super common method these days, but it was still pretty new at the time. This process let them compare their hybrid plasmid with plasmids that they knew had the gene of interest, right? They're checking that the right gene made it into the new plasmid. And by March, they'd succeeded. They recombined the gene into a new plasmid and put that plasmid into a bacterium. But that wasn't all. Those bacteria then replicated, which meant they'd also created clones, or bacterial offspring with the same genes as the parent. Cohen and Boyer's first paper was finally published in November of 1973, but in the time between finishing the previous experiment and publishing the paper, they started a few other projects. Their first experiment involved splicing two E. coli genes. So taking it a step further, Cohen's lab at Stanford decided they'd try to combine plasmids from two different bacterial species. And when that worked, the gears started turning even faster. Could they splice a gene from a more complex animal into E. coli and then get the bacterium to make a specific protein? But before I explain that, I should probably explain what make a protein means, right? Protein isn't just the stuff in food. Proteins are a broader class of chemicals that include everything from hormones to enzymes. And proteins are made of smaller pieces called amino acids. Some proteins only have a few amino acids, while some have thousands. Now, inside your cells, there are little organelles called ribosomes that gather these amino acids and put them into a sequence, and that becomes a protein. The cell knows which amino acids to grab because of DNA, or deoxyribonucleic acid, which is like a set of genetic instructions. I use the analogy that DNA is like a cookbook, and each gene is like a recipe for a specific protein. Now, ribosomes can't read DNA, so a bunch of enzymes have to come in and create a mirrored version of DNA that it can read. This is called RNA, or ribonucleic acid. So the big takeaway here is that DNA is a set of instructions for gathering amino acids and building proteins. DNA makes RNA, makes protein. But here's the coolest thing to me. With a few exceptions, like in certain mitochondria, every species' cellular machinery interprets RNA as instructions to grab the same amino acids. To use the cookbook analogy, every living thing has a different cookbook, but the recipes are written in the same language. This is what made Cohen and Boyer's third experiment an even bigger deal. They inserted DNA from a frog into bacteria. The bacteria successfully took up the new DNA and passed it to their offspring. They published their article in the Proceedings of the National Academy of Sciences in May of 1974, and it got media attention right away. An article in the New York Times suggested that this kind of technology might be used to create drugs like insulin. Now, Cohen worked at Stanford, which at this point becomes a crucial part of the story. And that's because Stanford had recently started a department called the Office of Technology Licensing. So whenever their faculty invented something new, the office could help them get a patent and sell licenses. Then the royalties from the licenses would help fund research and fund the greater university. So in 1974, when the head of the department read that New York Times article, he suggested that Cohen apply for a patent. But that was a dilemma for Cohen and Boyer, because there was still a stigma around patenting academic discoveries, especially in medicine where inventions could benefit patients all around the world. It definitely wasn't unheard of, but getting a patent would come with criticism from their peers. Plus, their research had received government funding from the National Institute of Health, or NIH, so neither Cohen nor Boyer thought they'd even be eligible for a patent. Cohen was especially worried about the ethics. According to a story he wrote 40 years later, quote, "My first reaction was quite negative. Could findings of basic research funded by the public be patented? And should they be?" The head of the Office of Technology Licensing talked to Cohen and told him that if they did file a patent, it would get the technology out to other researchers more quickly. And contrary to what they thought, getting NIH funding didn't automatically disqualify them from applying for a patent. And as far as money went, they'd only make businesses pay royalties. Cohen ultimately went along with the patent, but declined his share of the royalties. Boyer was on board too, and on November 4th, 1974, they submitted the first version of their patent, Process for Producing Biologically Functional Molecular Chimeras. Now, while they're working out the details for the patent, scientists around the world were getting anxious about the technology. There had been safety meetings and open letters before, and even a call for a moratorium on recombinant DNA research. It was signed by Berg, Cohen, and Boyer, among others. But this came to a head in February of 1975, when over 100 molecular biologists got together at Asilomar State Beach, right next to Monterey, California. They were there to talk about the state of recombinant DNA. If they were going to bring this technology into the world, they wanted to make sure they did it safely. And by the end of the conference, the general vibe was that they needed some rules in place to say what scientists could do with recombinant DNA. The NIH had already put together an advisory committee to do exactly that, but a little while after the Asilomar conference, the NIH wrote down some more explicit rules that scientists would need to follow if they wanted NIH funding. They went through a bunch of different versions of the guidelines, with some scientists saying that they were too lax and would lead to something bad happening, while some said that the guidelines were too restrictive and would slow down progress. All that's to say, biologists were taking recombinant DNA very seriously in the mid-70s. At the same time, though, so were investors. Okay, when people think of Silicon Valley, they think of technology businesses like Apple and Google. That's because back in the 70s, some venture capitalists started investing in semiconductor companies and made a lot of money. That attracted more venture capital to the area, which attracted more tech companies, and that cycle continued to the point where my hometown of Santa Clara is home to a $1 trillion company. We've got a handful of massive universities in the area too, which certainly helps introduce new ideas and inventions. Now, at this point in the story, that culture of entrepreneurship was just starting to gain steam. By 1975, tech companies like Intel and Atari had all gotten VC funding. So when investors started looking for the next big thing, they looked at this new field of biotech. Which brings us to our finance guy of the video, a 20-something-year-old investor named Bob Swanson. He was super interested in recombinant DNA technology, so after parting ways with a famous VC firm called Kleiner Perkins, he decided that he would start a biotech company. So he started calling the scientists who attended the Asilomar conference and eventually got a hold of Boyer, who agreed to meet with him. The two of them got along right away and talked about what a partnership might look like. Boyer didn't want to leave academia, but he imagined a side business would help him fund his lab, so he was in. But first they needed a product to sell, and after some thought, they landed on insulin for two reasons. One, it had business potential, and two, it was biologically feasible. From a business perspective, insulin was a good target because it was already a well-known drug with a global market of hundreds of millions of dollars. The number of diabetics was growing too, which meant so would the demand for insulin. Meanwhile, the insulin supply, which mostly came from pig and cow pancreases still, was predicted to flatten out in the 80s. So switching from livestock insulin to insulin made by bacteria would let producers keep up with demand. And from a biology perspective, insulin seemed within reach because they weren't starting totally from scratch. The sequence and 3D shape had been known for years. A molecule of insulin was made up of 51 amino acids, 21 in its A chain and 30 in its B chain. Then the two chains are held together by disulfide bonds. Pig insulin is similar, but differs by one amino acid at the end of the B chain. It's got an amino acid called alanine, where humans have one called threonine. And more recently, research groups in China, Germany, and the US had all figured out how to create synthetic human insulin in the lab. A research group in Switzerland then ran a super small trial with 6 diabetic patients and successfully treated them with synthetic insulin for 2 weeks. So human insulin had been made, but it was too expensive and time-consuming to produce on a larger scale. And that would be Boyer and Swanson's business advantage. By using recombinant DNA, they'd be able to turn bacteria into little machines that made human insulin. And that would be more efficient than the older production styles, both the lab-made human insulin that was extremely costly and the insulin extracted from pig pancreases, which was limited. Then once the process was dialed, they would license the technology to a pharmaceutical company who would bring human insulin to market. More on this later. So Swanson wrote up a business plan that made it seem like the technology was ready to go. He was doing the extremely Silicon Valley thing of exaggerating so that their business would seem like a good investment. But it worked. Swanson presented their business plan to his old employer, Kleiner Perkins, who promised them $100,000 in exchange for shares in the company. So on April 7th, 1976, Swanson and Boyer formally founded Genentech, a portmanteau of genetic engineering technology. Just one little problem. Boyer and Cohen assigned the rights to their patent to their universities, so Boyer couldn't automatically use it. And without a license to that patent, Genentech's business model wouldn't work. So Swanson went to Stanford's licensing office and came away with a non-exclusive license. While Swanson wanted a more exclusive license at first, Stanford wanted other institutions to be able to use the patent, so they went with a non-exclusive license. Awesome. Now Genentech needed a lab. And while Boyer's lab at UCSF would be convenient, they received funding from the NIH, and Boyer didn't want to mix company research with government money. So in August 1976, Swanson struck a deal with the university. Genentech would fund Boyer's lab in exchange for exclusive licensing rights to any new recombinant DNA products created as a result of the funding. So private funding wouldn't cross streams with public funding. Finally, NIH guidelines said that if you studied DNA from a human source, you'd have to use special high-security research facilities. Private companies didn't have to follow the guidelines, but Genentech followed them anyway. Now, there was an exception to the NIH guideline. If you studied synthetic DNA, you could use a lower security lab, and Genentech knew a facility that would be perfect to work with: the City of Hope Medical Center near Los Angeles. They were one of the only facilities around who could synthesize DNA. Plus, Boyer had collaborated with 2 of their scientists before, Arthur Riggs and Keiichi Itakura. He could vouch for the quality of their work. Now, Boyer proposed starting on insulin right away, but Riggs and Itakura weren't as confident. Instead, they proposed an experiment with somatostatin, a smaller, simpler protein just 14 amino acids long compared to insulin's 51. There wasn't much of a market for somatostatin, so this was more of a proof of concept experiment than anything else. Boyer agreed and said that Genentech would fund it, so Swanson raised a bunch more money, including another 100 grand from Kleiner Perkins. Somatostatin research with City of Hope would finally start around February of 1977, but it was far from simple. One of their biggest challenges was getting E. coli to read the entire gene. So even though somatostatin is small, the team decided to make the gene in smaller pieces. City of Hope started by creating the synthetic gene in fragments, then they'd ship them up to San Francisco, where Boyer's lab tried to figure out how to attach the fragments together and clone the gene. Then they'd send it back to LA, where City of Hope would see if any somatostatin popped out of the bacteria. And at first it didn't work, but then the scientists realized something. There was a chance the cell was making the protein, but enzymes in the bacteria were breaking it down before the scientists could detect it. So they pivoted to a totally different strategy. If they could make a gene that coded for somatostatin plus a big protein tail, maybe that would let it survive the destructive enzymes. And it worked. In August 1977, just 6 months after starting, they successfully used an artificial gene to create functional somatostatin in a bacteria. They published their results in Science in December 1977. So now that they'd shown they could make human proteins with recombinant DNA, Swanson decided it was time to invest in their own facility. They bought a warehouse in the city of South San Francisco, which is the biotech hub of the Bay Area these days. You'll actually see Genentech's name on a sign welcoming you to the city when you get off of Highway 101. But once again, this is a business, so they needed even more money to cross the finish line. Swanson knew he couldn't keep tapping VCs for money, but if he could partner with a large pharmaceutical company, that might give Genentech more consistent and dependable funding. Plus, if they did it right, they wouldn't just get money, they'd get access to a team who knew regulations and the FDA approval process. The ideal partner would be any of the 4 main insulin producers in the world at the time. Eli Lilly was the biggest with about 40% of global production. They were followed by Novo with 25%, then Germany's Hoechst with 8%, and Nordisk with 5%. Also, once again, I'm sorry to the German speakers out there, it is what it is. They talked to Novo and Hoechst first, but couldn't strike deals with either, so they turned to the American companies. Eli Lilly sold the vast majority of insulin in the US at the time, while a smaller company called Squibb made up most of the rest of the market. So Genentech met with Squibb first, thinking that they'd see recombinant DNA as a competitive advantage over Lilly, but Squibb's consultant didn't think the product would work, so they said no. Lilly, on the other hand, was looking for something new. Cow and pig insulin sometimes triggered allergic reactions in humans, so they wanted a version that wouldn't cause those issues. But recombinant insulin would also fix their supply problem. It took a lot of pig pancreases to make a dose of insulin. Recombinant DNA in bacteria would give them a renewable insulin factory. Making things more urgent, though, a research team at Harvard were using a different type of genetic engineering technology to make rat insulin. Their team's main researcher had co-founded a company in Switzerland called Biogen with the goal of producing and selling human insulin. Plus, the Danish companies were planning to enter the US insulin market soon. So after learning about Genentech, Lilly's head of research took a meeting with Swanson. Lilly was interested in the technology, but they knew there were still risks. So they hedged their bets in a few ways. At first, they would fund Genentech's insulin research at $50,000, and then when they met certain criteria, Lilly would increase the payments. With funding secured, Genentech scientists got to work. Their plan was to synthesize genes for the A and the B chains separately, then combine them after the bacteria had produced each chain. But joining the 2 chains together was a big challenge. Luckily, after a bunch of failed attempts, they figured out that the B chain was contaminated with proteins that kept it from linking to the A chain. Shortly after realizing that, Dave Goodell, a researcher they brought on from the Stanford Research Institute, successfully joined the 2 chains, which meant that on August 21st, 1978, they'd done it. Scientists created the first synthetic human insulin made with recombinant DNA. They published the paper, Expression in Escherichia coli of Chemically Synthesized Genes for Human Insulin, in the January 1979 issue of the Proceedings of the National Academy of Sciences. Now, if this were purely academic, I'm sure the team would have celebrated for a week. But this is industry, so the 2 businesses didn't waste any time. Within a week of the successful insulin experiment, Lilly and Genentech negotiated a 20-year research and development deal. Lilly would pay $500,000 lump sum in return for exclusive global rights to Genentech's technology that made human insulin. On top of that, Genentech and City of Hope got a small percentage of the product sales as royalties. But Lilly still needed to be careful in case the production process didn't scale like it was expected to. So they stipulated that the Genentech scientists would still have to hit certain research milestones by certain dates in order to get paid. Meanwhile, Lilly had a similar contract with a different research group at UCSF. If one technology didn't work out, hopefully the other one would. Also, yes, in the middle of all this, there was another group at the same university as Boyer doing similar research. There's so much more to the story that I couldn't get into in this video, so I highly recommend the 2011 book Genentech, The Beginnings of Biotech by Sally Smith Hughes. She conducted firsthand interviews with so many of the people from the story. I could not have written this script without her work. I'm hugely grateful to her. I'll have a bunch of her sources down in the description and a link to the book as well. Thank you, Dr. Hughes. Either way, now that human insulin from recombinant DNA was possible, it was time to scale up production. And what they wanted to do was follow the example of beer brewers and antibiotics manufacturers by using big fermentation tanks of bacteria. Unfortunately for them, existing NIH guidelines said that you could only grow this kind of bacteria in 10-liter batches max. But in January of 1980, after a bunch of lobbying, the NIH director lifted this limit in certain situations. And with the new relaxed rules, Lilly was able to build 2 new factories and make enough insulin for clinical trials. One of the first trials happened at Guy's Hospital in London and compared the safety of recombinant insulin with insulin from pig pancreases. They confirmed that in 17 healthy participants, recombinant insulin was as safe and effective as animal insulin. Eli Lilly broke the news publicly in July of 1980, and the results were published in The Lancet in August of 1980. And since Lilly was already a pro at insulin trials and FDA applications, the Genentech Lilly insulin got approval on October 28, 1982. Lilly released it under the brand name Humulin, making it clear that this was human insulin. After the 1979 paper, Genentech would go on to have even more business success. Recombinant DNA would allow them to make proteins and hormones more efficiently than before, which pharma companies were willing to license. The Genentech story showed investors that biotech was indeed the next big thing. Okay, so at this point in the story, human insulin has hit the market. But like I mentioned in the intro, over 90% of America's insulin is another product entirely, which doesn't mean that human insulin was a waste of time, just that it was a stepping stone. While the American insulin market was excited about recombinant insulin, the Danish producers, Novo and Nordisk, weren't as into it. According to Pierre de Mates in 2017, some of Novo's scientists even referred to this new recombinant insulin as, quote, sewer insulin. Plus, Novo already had a popular, highly purified animal insulin called monocomponent insulin, so they weren't in a rush to develop a whole new technology. But despite their initial resistance, it was clear that human insulin would become more popular. Like, given a choice between pig insulin and human seems better. This pushed Novo to pursue their own human insulin product, but with a totally different technique. They used a method called transpeptidation, literally transferring peptides, or chains of amino acids. Remember how human and pig insulins are nearly identical except for that one amino acid at the end? Well, scientists took insulin from pig pancreases and used enzymes to swap out the alanine from pig insulin with threonine, making it chemically identical to human insulin. The final product became known as humanized insulin, or semi-synthetic insulin. Novo released it in Europe as Novolin in 1982, and actually beat Lilly's Humulin to the European market. Hoechst followed in 1983 with their own humanized insulin products in West Germany and Austria. The last of the major players to get involved then was Nordisk, but they weren't convinced that human insulin was any better than purified pig insulin, since that one amino acid difference shouldn't have affected how well insulin lets glucose into the cell. Human insulin was certainly marketed as better, but Nordisk was skeptical. The evidence was building throughout the 80s, but I'm going to skip to a 2005 Cochrane review. These are high-quality systematic reviews in the health sciences. And the 2005 review asked whether human insulin could control blood glucose better and lead to fewer adverse reactions than purified animal insulin. They analyzed 45 randomized controlled trials that had come out since the invention of human insulin, summarized the data, and concluded that there weren't, quote, clinically relevant differences between the two. This meant that the last 2 decades of research and the millions of dollars of investment hadn't actually created an insulin that was better for the patient, just an insulin that sounded better. The research did make something better possible, though. See, at this point in the story, diabetes management used something called the basal and bolus approach. Diabetics would get a long-lasting basal insulin that would stabilize their baseline blood glucose, especially overnight. Then, when it was time to eat, they'd supplement with a bolus, or a dose of fast-acting insulin to deal with that rise in blood sugar. It's similar to how a healthy pancreas releases insulin. So most insulin makers had at least 2 insulin products. They'd have long-lasting insulin for the basal dose and fast-acting insulin for the bolus. And chemically, the difference between the two comes down to how quickly the insulin breaks apart after it's injected. See, single insulin molecules group together in pairs and in groups of 6, kind of like slices of a pizza. This 6-slice insulin pizza is organized around zinc, which keeps it from breaking apart right away. This is why old-school drug developers added zinc to long-lasting insulin formulas. But it's those individual insulin molecules, those single slices of pizza, that actually interact with insulin receptors on our cells and start the process of bringing glucose into the cell. So when drug developers were trying to perfect the basal and bolus formulas, they tried to optimize either how well the insulin pizza stayed together or how quickly it dissociated into single slices. And recombinant DNA might give them a way to do that. Their first target was a better mealtime insulin, one that broke apart more easily. And scientists thought that if they could replace the amino acids on insulin that bound to zinc, then the 6-piece insulins should be able to dissolve into single molecules of insulin faster, which would mean a faster acting insulin. Unfortunately, some of the first attempts didn't go so well, like some led to tumors in rats. But by 1996, Eli Lilly had found a winner. Normal human insulin has a section at the end of its B chain where a lysine sits next to a proline. So Lilly used recombinant DNA to design a version where they swapped positions. And the insulin worked as expected. The insulin slices weren't held together as strongly, so they dissociated into single slices more easily. Since it involved lysine and proline, they called it Insulin Lispro, which they sold under the brand name Humalog in the States. And they named it that because Insulin Lispro is what's called an analog to human insulin. An analog is a substance that has a similar effect on the body and or a similar structure to another substance, so insulin analogs do the same job as insulin. These analogs are the majority of insulin products used in the US today. Okay, so Novo and Nordisk merged into Novo Nordisk in 1989 and created their own insulin analog. They replaced the proline amino acid towards the end of the B chain with aspartic acid, so this one became known as insulin aspart. They sold it as Novo Rapid in Europe in 1999 and Novolog in the US. Now, while Novo Nordisk and Eli Lilly focused on fast-acting insulin analogs, Hoechst, who had merged with another company to become Aventis, used a different strategy. They wanted to make the first long-lasting insulin analog. It was called insulin glargine. For this one, they replaced the typical asparagine at the end of the A chain with the amino acid glycine, which gives us the GL part of the name. But they also added 2 arginines at the end of the B chain, which gives us the argine part. So voila, insulin glargine, the first slow-release insulin analog. Aventis sold it under the brand name Lantus starting in 2001. And since it was the first long-lasting insulin analog, it became the best-selling drug in the category for years. And in 2004, they got FDA approval for their fast-acting insulin glulicine. This one has a glutamine substituted towards the end of the B chain and a lysine sub towards the beginning of the B chain, hence insulin glulicine. Later that year, the French company Sanofi acquired Aventis and they became Sanofi Aventis. I know there's a lot of business in this episode. For those keeping score, Sanofi Aventis had 2 analog products while Lilly and Novo Nordisk only had their fast-acting analogs. So Novo Nordisk decided that they'd try to compete with Lantus by making a long-lasting analog of their own. They called it insulin Levemir, and while I couldn't find a source for this, the name probably comes from the myristic acid they added to the end of a modified B chain. I'll talk about this at length in the next video about GLP-1s, but Novo Nordisk found that these fatty acids could get molecules to bind to albumin in the blood serum, which created a chunky molecule that stayed in the bloodstream for longer. Insulin Levemir got FDA approval in June 2005, and Novo Nordisk released it as Levemir in the US. Meanwhile, Eli Lilly still didn't have a long-lasting insulin analog of their own. Instead, they decided they'd just make generic insulin glargine when Sanofi's patent expired in 2014. Lilly sold it under the brand name Basaglar in the US. They calculated they'd be able to sell it for about 20% cheaper than brand name Lantus. Then in the 20-teens, Novo Nordisk came out with another insulin analog, insulin degludec. Compared to insulin Levemir, it had a longer fatty acid chain, lasted longer, and only required 1 shot per day instead of 2. Okay, so I know I just sped through like 25 years of drug development, but it's all for the sake of showing an overarching trend. The modifiability of insulin analogs replaced human insulin, which replaced animal insulin. A study published in 2011 by Gill et al. Compared the insulin products used around the world from 2000 to 2008, and it pointed to the same trend. By 2008, very few people around the world were still using animal insulin anymore, but a country's income predicted how much they used analogs versus human insulin. The analogs are more expensive, so insulin users in low-income countries used mostly human insulin and used the most animal insulin. Meanwhile, high-income countries used mostly insulin analogs and a smaller percentage of human insulin. Some of these trends must have influenced Sanofi, who, at the end of 2019, announced that they were quitting diabetes research in favor of more profitable areas. Their insulin glargine, Lantus, was still popular, but revenue was declining. And while they'd been working on new diabetes products, they couldn't quite land on something new to bring to market. And in 2023 and 2024, Novo Nordisk made similar announcements. They'd stop selling Levemir in the US, but still sell insulin icodec, a once-a-week basal insulin in approved countries. They weren't stopping insulin production, just scaling back certain things. Plus, some of their other diabetes drugs were taking priority. More on that in the GLP-1 video, which meant we'd come full circle. Scientists spent all that time and money to develop new human insulin, only for the biggest insulin makers to pull back 40 years later. So what's the takeaway here? Well, number one, recombinant DNA's biggest immediate impact was efficiency, not new drugs. See, while human insulin wasn't necessarily better, the new technology allowed drug makers to fix their supply problem. So instead of being limited by the supply of pig pancreases, insulin makers could use virtually unlimited E. coli to manufacture insulin. And this kind of tech could be applied to other proteins, one of which was a Genentech follow-up product. See, Herb Boyer's son suffered from stunted growth, and for a while, doctors thought his son would need to take growth hormone. But in order to get any, a drug manufacturer would need to mush up pituitary glands from cadaver brains. And just like the pancreas supply problem, there's a limited number of spare pituitary glands out there, which makes the hormone hard to get. So Genentech created recombinant growth hormone in E. coli and partnered with a Swedish pharma company called Cobigen, who mass manufactured it and made it available for patients who needed it. Just like human insulin, the breakthrough here was the efficiency of production, not the effectiveness of the product per se. Takeaway number 2. The relationship between Genentech and Lilly became a model for how biotech and pharma could work together going forward. That's because each industry has something that the other one wants. Biotech companies learned that if they developed a new technology, they could license it to pharma companies who had the resources, like money and infrastructure, to conduct clinical trials. Then the pharma companies could scale up production and bring the drug to market, something that would be too expensive for a small biotech startup to do on its own. And from pharma's perspective, biotech companies handle the early stage work that's often too risky for pharma companies to invest in directly. What the industry learned from Genentech's origin story was that if they backed the right company, they might get access to a novel technology that gives them a competitive edge. So instead of pouring money into their own in-house R&D, which can be both expensive and risky, they could just acquire innovation from biotech firms. So biotech companies get the funding and scale that they need, while pharma companies get the novelty that they need. Finally, takeaway number 3. Recombinant DNA did eventually lead to better drugs. If you knew the protein's amino acid sequence, you could create synthetic DNA and print it on demand. And that worked well for drugs with known blueprints like somatostatin, insulin, and growth hormone. But it also allowed drug developers to tweak those drugs into analogs with different properties. They could build a better bolus insulin by modifying specific amino acids and creating analogs. Speaking of which, a different molecule's analog became super buzzy a few years after the insulins. It was an analog of glucagon-like peptide 1, a molecule that could control diabetes, but also, it turns out, help its users lose weight. In the next video, I'll tell you the fascinating backstory of GLP-1 receptor agonists like Ozempic and Wegovy. But real quick, these videos take a ton of work to pull off, and if you're in the position to support my work, consider heading over to my Patreon and pitching in a couple bucks a month. I got links here and down in the doobly-doo. Take care. Thanks for watching.